Okay, we're ready to go. There are 4 handouts in the front here from me, today's lecture, and the next 3 after that, so make sure you've got them, okay, by the end of the class. All right, if everyone wants to sit down, and I'll start talking. Okay, today begins bacterial pathogenesis, and what we're going to be doing is giving an overview of the sorts of things that can happen in a bacterial infection and infectious diseases. Obviously, we aren't going to cover the whole of bacterial pathogenesis in all aspects in one lecture. What we're going to be doing is giving examples and sort of things you should be looking at as you look at the individual diseases. I've put at the end of this handout 2 summary tables, and I specifically set them for quiz 4 review. This is not going to be on the next quiz. It's ridiculous. These 2 tables. It's ridiculous to just to memorize a bunch of names when you don't know what they are, and we obviously haven't had the individual organisms yet. So this is a summary you can look at before you start or any time during, as you look at the individual organisms, and at the end for revision for the next quiz and for the final exam. And similarly, I said some of the major exotoxins. I've put down pretty well all the ones that I cover. There's a few I've missed out that other people cover because I don't want to have 2 sets of information here, 2 different perspectives. So if you find it useful, you might want to add in some of those. Yeah, sorry? Can't hear what? Oh, okay, that's fine. Good idea. Okay, so anyway, that's the 2 types. So don't, you can look them over. They'll be, because we're going to be, we're starting the individual organisms next lecture. So these 2 overview things will be useful, but you don't have to memorize them for the next quiz. Okay, you've all been watching the news and the newspapers, the TV, what have you, the last few days, and you've come across spinach disease. Okay, I mean, every year there's some variant. Sometimes we get a new disease caused by a new type of strain, a new species. This is nothing magical in microbiology. Every year this happens. This year it's caused by, presumably, the enterohemorrhagic E. coli that is found in human feces and animal feces and what have you, and probably got on the spinach. We'll have a lecture on Enterobacteriaceae later. The only reason I bring this up is but you can't learn everything in bacteriology because it changes every year. What you can do is get the basic understanding and know how to adapt as nature adapts and as new things happen. Okay, so let's get into pathogenesis. First of all, there's the general aspects of bacterial pathogenesis. That's what we're going to be doing. This is broken down into 3 sections today, really. First of all, general terms that relate to just the language that we use in medical microbiology and infectious diseases. The processes that can occur in a bacterial infection, and no, they don't. All of these things don't happen in every disease in the same way, because if they did, there would be no reason to study different diseases. We just say, well, they'll work this way, and they don't. So these are the sorts of general processes that can happen, and as we get into the individual organisms, the individual species and strains as they cause very specific diseases, later on we will be basing it on these concepts. And then we're going to talk about a few terms here which relate to the ways that bacteria can damage or kill you, and of course we have a new term since 2001. And there'll be 3 lectures on that by Dr. Ghaffar. I just put in a couple of paragraphs relating to this because I've had a long-term interest in this for about almost 30 years now. Okay, so let's move on. Pathogenicity. All right, first of all, individual strains within a species or a whole species in some instances may have particular virulence factors. Okay, so we need to know not just what these factors are, but to understand sort of how they relate to a particular disease. Some organisms are they just require very few organisms, and some diseases require many. And we don't generally, in microbiology, sit down and define exactly how many organisms it takes to infect a human being, because that would be impossible to do if you think about it. But anyway, unless anybody wants to volunteer today to come down with hemorrhagic fever or maybe rabies or something. But anyway, so it's just not something that, in bacteriology or virology, we generally do this sort of thing. But we do have a sort of a general idea. But in terms of understanding pathogenesis, it is important. Because an organism that for example, let's take gastrointestinal infections. There are very few Enterobacteriaceae, which are gram-negative facultative anaerobes. There are very few organisms, Enterobacteriaceae, in the gastrointestinal tract. Yet, if you, for example, have most of the organisms, in fact, strict anaerobes in the intestine. But if you, for example, receive a knife wound and through the gut, and the stuff perforates into the peritoneum underneath, if the chances, many times you won't get an infection with a strict anaerobe, you'll get an infection with a facultative anaerobe. And that's an example. It isn't just an issue of the number of organisms that you are exposed to. It's their potential to take off and grow and cause disease in you. The immune status is also extremely important. If a person is immunocompromised, their immune system is not just an issue of whether you were exposed to antibiotics or not. The immune system is extremely important in stopping people getting infection or infectious diseases. And so an immunocompromised person, somebody on cancer chemotherapy, as an example, would be much more susceptible to a bacterial infection than a normal person. So these are the sorts of things in the back of our mind we're thinking about in general as regards an infectious disease and the organisms that cause it. Now, in fact, particularly in a hospital, you don't see sort of a lot of times epidemics with what we call primary pathogens. The big deal in a hospital, for example, would be an infection with an opportunistic infection or an organism that really doesn't really do much to healthy people. And so you don't want to think about as pathogens as being the big deal. They are a big deal, but not the big deal. In many situations, and again, such as a hospital as an example, the big deal are the opportunistic infections, the organisms that don't cause disease in healthy people. And of course, many of you, at least before you go into general practice, will all of you be working in hospitals, and many of you will continue to be associated with hospitals. And so, these are definitions, but we don't understand what we're saying. So yes, pathogens cause epidemics in healthy people, and they are very important. But equally important are opportunistic infections, which we'll get to in a second. Now, Koch's postulates were written about 100 years ago by Koch in terms of what he knew about bacteriology and what the world knew about bacteriology and virology at that point, particularly bacteriology in his case. So this is not strictly Koch's postulates, but it's a slight sort of revision on them. So I don't want anybody to say that I'm quoting word for word what Koch said. Again, this isn't a history class, this is a microbiology class, okay? So this is basically a slight adaptation of Koch's postulates. First of all, isolating the organism. We must isolate the organism from disease, but not healthy people. Secondly, you should be able to grow the organism in pure culture. And finally, you should induce the disease in susceptible animals. And finally, they should be re-isolated from susceptible animals. Well, unfortunately, even in the year 2006, we don't have animal models for all infectious diseases, and we can't even culture some bacteria in the laboratory. So if we strictly stuck with Koch's postulates, we'd say that, for example, that syphilis is not an infectious disease of human beings. Of course, it's the number 2 sexually transmitted, within the top 3 anyway, infectious diseases in the country, depending how you look at it. And certainly, it doesn't meet Koch's postulates, but that certainly doesn't mean it's not a human infectious disease. So we need to understand what Koch was getting at, but we don't necessarily have to sort of look at every disease the way that things were looked at 100 years ago. But this is what we'd like to happen. We'd like to be able to meet these criteria. Isolation from healthy disease, not healthy people, well, that's fairly straightforward. We'd like to be able to grow the organism in pure culture. Most of the time, that's not a problem. But as I've said already, a few organisms cannot be grown in culture, period. And if we'd like to induce them in susceptible animals, we'd like to re-isolate. Well, if we don't have a decent animal model, we don't have an animal model that looks anything like the human disease, well, clearly we can't carry out 3 and 4. Okay, so this is what postulates are all about. Opportunistic infections. Okay. Opportunistic infections are basically from what happens in compromised people. People who've got some reason that they get infected more easily by bacteria or certain bacteria than other people. The terms that you should know about, the important term is the normal flora. The body surfaces are covered in bacteria. They are absolutely chock-a-block covered. Most of the weight of fecal matter is actually bacterial in nature. The lungs, all of the external surfaces are absolutely chock-a-block covered in bacteria. That's the normal flora. Now, clearly, the normal flora are not a problem most of the time. If they were a problem, we'd all have bacterial diseases. We'd all have infectious diseases 99% of the time, or we'd be all on antibiotics all the time. So clearly, the normal flora aren't a problem most of the time. But they are a problem when things go wrong. Minor things sometimes happen, like boils caused by Staphylococcus aureus. Major things can happen, such as I've mentioned already, situations where the intestinal tract in particular is perforated in one form or another. It could be a natural perforation, as well as a man-made one. Usually we talk about the environment, and people who really don't know anything about bacteriology sort of really get totally the wrong end of the stick. We are constantly exposed to bacteria. This air is chock-a-block full of bacteria. You are breathing it. We can culture very easily bacteria from the air that you're breathing at this moment. It's not a problem. But again, nothing happens. Because most environmental bacteria don't do anything. They don't sort of cause human disease. There are primary pathogens, such as Legionnaire's disease, Legionella pneumophila, that lives in hot water towers. Bacillus anthracis is an environmental organism that lives in the soil. They're both pathogens, not opportunists. There are other organisms, such as Pseudomonas aeruginosa, which normally does absolutely nothing to normal people. It is an environmental organism that's water-loving. But if you happen to get, for example, some saline that's given intravenously into a patient that happened to be contaminated, which is very rare, but if it were to happen, you could have a big problem. But what happens all the time is that patients, for example, who've got cystic fibrosis, who have major lung injury going on, are very susceptible to infections with Pseudomonas aeruginosa, which does absolutely nothing to healthy people. So, the normal flora certainly is an issue, and so is the environment. But we should realize, again, even in these opportunistic situations, that some bacteria just happen to be able, for whatever reason, and so we will define some examples as we go on, to cause infectious disease, and other organisms just don't. Okay? So we shouldn't just say even though it's opportunistic, it's not a totally non-specific sort of thing where anything can happen. Some examples of opportunists in the normal flora. The skin. And there are many, many different bacterial species found in human skin. And I'm sure, and I'm quite confident that every one of you would have quite some differences from everybody else in the sorts of flora that you find. Although this is something that basically is almost never looked at. But what we do know is some species crop up again and again. For example, Staphylococcus aureus is a particular interest because it can cause quite nasty opportunistic infections, which can spread into the bloodstream. It can infect just about any tissue. Although most of the time in a normal person it does absolutely nothing. Staphylococcus epidermidis is another organism, and we will have much more detail on this, for example, in the Staphylococcus lectures that I'll have later. Propionibacterium acne is one that's particularly a lot of attention is paid to as a contaminant because it can be confused with Corynebacterium diphtheriae if one's not very awake. In the intestine, among the many different types of obligate or strict anaerobes which are present in the intestine, Bacteroides in particular particular Bacteroides fragilis, causes many cases of opportunistic infections compared to other organisms that are present. And in this instance, I'm contrasting Bacteroides versus Enterobacteriaceae. I'll rewrite this thing a little bit when I get to the, or restate it again, when I'm dealing just with strict anaerobes, comparing Bacteroides among other strict anaerobes. But at the moment, I'm comparing this with a Bacteroides with a strict anaerobe, specifically with the Enterobacteriaceae, which are facultative anaerobes. And if you wonder why I'm saying a lot of words that you're not totally familiar with, I remind you again, we've had the first 5 lectures, we've had this overview, and you need to learn this language because we're going to be using it now. Okay? So we don't differentiate the clinical part of the course here as basic language from the basic part of the course. Okay, so these are facultative anaerobes and these are strict anaerobes. We will have a whole lecture on, 2 lectures on Enterobacteriaceae and 1 on strict anaerobes. These are just examples today to give you a taste of what's to come and how to start thinking about things. Okay? Opportunist environment. Yes, they can come from the air, the water, soil, or the food. And that relates to particular situations of, particularly think of in a hospital situation of what might happen and how you need to stop it happening. And that relates to infection control in hospitals. Okay, opportunistic infections in hospitals are so common. Many people die each year in hospitals, not because they walked in with the disease that they started with, but they were in such bad shape. They wouldn't be in a hospital if they weren't in bad shape. But they're much more susceptible than the average member of the population. And it's so common that it's referred to as nosocomial. And that doesn't mean that hospitals are sort of hotbeds of disease and death. It means that anybody's doing anything wrong. It means that these people are, as we said already, they are compromised in some fashion, or immunocompromised specifically, and they're set up and they're ready. And no matter how good a job you do, people are going to get infections in hospitals. And that's why we use this special term, nosocomial, to emphasize how common it is. Okay. Transmission. All right, now what we're going to do now, we come up with some general terms relating specifically epidemics caused by pathogens and opportunistic infections relating from the normal flora, from the environment. Go ahead. Right. Yes.
Are there more opportunistic bacteria in the hospital, or would any compromised outside the hospital?
Well, that's a good question. That's a good question. Take, for example, I don't think there was more as a general, just an absolute load. But take an example of MRSAs, methicillin-resistant staphylococcus. If you happen to have a patient who has such a disease for whatever reason, it could easily spread to others in that ward, and you could end up with a whole ward full of a certain infection. So I don't think that, and so I think the argument is that what will happen is that because the people are already in bad shape, they will have the ability to get infected with a particular organism that might not normally be there, and that particular strain or species might be there in larger amounts or in a position to be transmitted, for example, on hands or from shedded skin from one person to another, which wouldn't happen. But the bacterial load is not higher. If the bacterial load's higher, then the hospital should probably close down and the people that run it fired, because we have disinfectants, we have everything else to clean hospitals. So there's definitely not. It's not just an issue of load; it's the issue of the circumstances. Okay, does that answer the question? All right, that's a good question. We're living in the real world, not in medical microbiology exam world, right? Okay. All right. Now the different steps in the process. All right. Again, we're talking generalities. We will talk specifics when we talk about individual organisms. There are various ways that things can be transmitted. For example, tuberculosis is primarily transmitted, but through airborne droplets. We've already been hearing about E. coli and spinach as an example in food. The water is often contaminated in the underdeveloped world with diseases such as Vibrio, which causes cholera. Sexual contact, there are specific organisms. And the point I'm making here is specific. The word is specific. It isn't just chance that some organism is transmitted through the air. It isn't just chance it's transmitted by sexual contact. There are reasons, there are rationales. The organism has got particular receptors or components or abilities that make it that it can do these specific things. So take an example, which is, of course, has been in the news more than it needed to be for the last 5 years now. There are very few organisms that form spores, as I've mentioned already. Many of them are Clostridia, which are strict anaerobes, which, as I've said before, are killed by the air. The only common aerobic organism, that is to say, an organism that survives in the air and that is spore-forming among bacterial pathogens, is Bacillus anthracis, causative agent of anthrax. And that's why it's transmitted, not necessarily through airborne droplets, but in the air. Okay, so there are reasons for this. There are specific reasons. It isn't just chance or, what had something vague. And we may not know the answers in every case, but in many cases it is known. Now, in terms of once an organism's got to a human being, the next thing is what happens. Well, there are many, many non-specific things that the human body is adapted to deal with the possibility of an infection. For example, peristalsis and defecation are ways of removing bacteria from the intestinal tract. The respiratory tract has ciliary action, coughing and sneezing as a way of trying to remove things from the respiratory tract. The urogenital tract, urination is a way of washing out bacteria. But remember, bacteria pathogens are very well adhered. They're stuck. They don't want to be dislodged. As we'll talk about next, it's a process that's designed to avoid these processes. And of course, there's the non-specific immune system, which you covered in great detail earlier, and I don't wish to or choose to do again. Okay, so adhesion is very important. There are often specific mechanisms by which a bacterial cell, a bacterium, and that's the singular, if we haven't emphasized, people have a horrible habit, and I probably do it myself because I'm not perfect. Some have a habit of using bacteria when they mean bacterium. The singular of bacteria is not bacteria, it's bacterium. But anyway, a single bacterial cell being shown here. And there are adhesins that are on the surface of these bacteria. And they interact with receptors, for example, on epithelial cells. That's a specific adhesin receptor interaction. And there are examples. I'll just give us a couple of examples as we go along. Take, for example, Streptococcus pyogenes. Again, here's a case where we see the epithelium, and we see fibronectin, not fibrinogen, fibronectin stuck on the surface of these epithelial cells. That's in this instance, would be the receptor, if you like. And then the Streptococcus has a particular protein called an F protein, or fibronectin binding protein, F protein. And lipoteichoic acid in the earlier literature was stated to be the adhesin. I've chosen for simplicity to just say that they're maybe both involved rather than get into a big debate on the literature. So this protein and lipoteichoic acid have both been suggested to be involved in binding to fibronectin. And that anchors the Streptococcus pyogenes, for example, to the pharynx in strep throat. I also going to be using, I've used the word before, fimbriae, and the singular for that is fimbris. And I really want to put this slide up here because I think it clearly emphasizes the difference between the fimbriae, which are these sort of hair-like structures that hang out from the surfaces of both gram-negative and gram-positive bacteria, as opposed to these whopping great things, these organelles, the flagella. Now, you wouldn't see this. Most of the slides that I show of this type are electron micrographs. You wouldn't see these fimbriae and you wouldn't see these flagella under light microscopy. But I'm emphasizing how different the fimbriae are, which are the adhesins, as opposed to the locomotory organelles, the flagella. For the purposes of what I'm going to be discussing today, I'm not going to be discussing the flagella. I will bring them up again as H antigens in the context of the Enterobacteriaceae and hamburger and spinach diseases. I know that's a silly term, I'm being facetious. But anyway, okay. All right, so fimbriae. And the reason I brought them as examples, here's an example of an adhesin that we see in gram-negative bacteria. And we see again the human cells underneath with the receptors and with the adhesin. Now in this instance, it's the bacteria that are recognizing the component on the host. In this instance, they can recognize some fimbriae from E. coli recognize mannose, and some will recognize galactose as a component of glycolipids and glycoproteins. It's common to find in eukaryotes as opposed to prokaryotes, post-translational modification leading to glycosylation is very common, which it is not in bacteria. And so it is very common for bacteria to recognize sugars, which are components of particularly of glycoproteins, but also of glycolipids that are present on the surface. And it's specific, because I remind you that mannose and galactose are actually isomers of one another. They're actually both 6-carbon sugars. And so it is that specific that these different fimbriae recognize different things. In fact, many years ago, Dr. Mayer spent a lot of his life working on these things, but he's forgotten that probably by now. Anyway, okay, so that's fimbriae. All right, now, another thing I want to talk about is penetration and spread. Most organisms cannot penetrate the human skin or any other. They cannot penetrate. As an example, it really is a very major barrier. But once it gets to the intestine or the respiratory tract, things are a bit different. Some organisms actually are in the gut lumen. They're along with all the food and other enzymes and what have you, and they're perfectly happy in that gamish. But they don't go any further. They stay on the surface of the intestine. Other organisms will actually get into the epithelium. And finally, some, and these are the worst of the lot, and that's why this enterohemorrhagic disease is such a problem, as we'll talk about in the Enterobacteriaceae lecture. If an organism is in your lumen, it can still kill you in the case of cholera. But it's a lot easier, and if an organism is stuck in the epithelium, it can still be pretty nasty. But once an organism gets into the lymphatics and the bloodstream, you're in bad shape. It's a hell of a lot harder to treat something when it's it's become a systemic disease. Okay? And that's what happens with these types of E. coli infections that are in the news at the moment. So that's what we need to know. Do they stay on the surface? Do they stick around and stay just within do they get endocytosed into the epithelium? Or do they actually go further? Now, the words penetration and spread are extremely vague, and people should not be confused. They should understand the process and not necessarily the words. If an organism penetrates into an epithelium, it's penetrating. If an organism penetrates into the bloodstream, it's still penetrating. Penetrating is not a microbiology term, it's an English term. Okay? So the issue penetration, what you need to know is understand the process. What do these things do? That's really the issue. And I've tried very hard to use specific terms. I've taught medical students for 26 years now. I try really hard to be specific in what I say. But sometimes the national boards are not so specific. And so the issue is that you need to understand the concepts and look at what's the question that's being asked, not just the word necessary that's being used. And spread can have different terms. It's just it's a term. It's not a microbiology term. It's English. These organisms will spread. But equally, well, one might use the term to talk about an organism spreading through a tissue. The point I'm making here is, where is the organism? Where is it? Where does it end up? Does it just stay put where it started? Does it get endocytosed into the epithelium? Or does it get further? But does it have the potential to become a systemic disease by spreading into the lymphatics and bloodstream and then ending up in all sorts of other tissues as a consequence? And we will discuss that. We'll give a good contrast, particularly in the Enterobacteriaceae and Vibrio lectures, because these three organisms will all be discussed at the same time. So that will make it, that's why I particularly pick these three, because it's easiest to point this out. There's a typo here I just noticed. Never mind. Connective tissue destruction. Okay, this helps bacterial dissemination. If an organism, for example, let's say somebody's got diabetes and they've got an infection of their foot and it's got very necrotic and nasty and what have you, if an organism produces collagenase to chew up connective tissue, the protein part, or hyaluronidase to chew up the proteoglycans, the human proteoglycans, as opposed to the bacterial peptidoglycan, which has got nothing to do with proteoglycans. If these things are chewed up, then the bacteria is more easily able to move through the tissue and get where it needs to go. Okay. Extracellular versus intracellular. We're going to give you some terms here and some examples, but the big issue is, it's not just an issue as does something live inside a cell, outside a cell being extracellular, or does something live inside a cell making it intracellular. The issue is what happens beyond that, and it really is an issue of the immune system. If an organism lives extracellularly and is quite happy living in the blood or the plasma, then the only thing it's going to come into contact with is antibodies and complement and lysis and other things, secreted proteins of that type, and particularly antibodies and complement. In fact, an extracellular pathogen isn't just chance again. Oh, it just happens to be extracellular. These organisms have lived with human beings for thousands of years, and they've adapted. We've tried everything we can to get rid of them, evolutionarily speaking. They've tried everything they can to avoid being got rid of. And so they're extracellular in many cases because these are organisms that are perfectly comfortable growing on just about anything, and blood and plasma is as good as anything else. But they've avoided being taken up by polymorphonuclear cells and macrophages. Indeed, these particular types of organisms, as they are taken up by phagocytes and polymorphonuclear cells or macrophages, phagocytes, they're usually killed because that's not where they want to be. Okay, so this is totally in contrast to an intracellular pathogen. Now let's take a couple of well-defined examples of bacterial substances that are known to be produced by extracellular pathogens that are involved in avoiding phagocytosis. One example is the protein A of Staphylococcus aureus. Now, I'm not going to, in this lecture today, nothing that I say, we will be saying these things again in terms of the specific diseases when we get into more detail later. Okay? So it doesn't say Staphylococcus aureus up there. You don't have to write down Staphylococcus aureus. I assure you, you're going to get again. And I'm sure it's also in the handout anyway. But what you do need to get out of this is what's going on. Okay, first of all, the bacterial cell produces this protein called protein A. Protein A specifically recognizes the FC region of immunoglobulin. Maybe I haven't shown it stuck in particularly the right spot. I don't know. I never claimed to be an artist. But anyway, it's stuck there, somewhere on that immunoglobulin, in that region. Now, what that means is what would normally happen is if an antibody response had been made against this organism, this extracellular pathogen, what would happen is that it should bind to an FC receptor and it should be internalized. But that doesn't happen. So this is a very well described, very well studied example of how an extracellular pathogen attempts to avoid being phagocytosed. Another, probably much more important example, there's no probably about it. We're going to, in our Streptococcus lecture, we're going to talk about the fact of M proteins being the site of vaccines against the group A Streptococcus, and why we don't use them, and why it's a problem. But anyway, the issue is that in a group A Streptococcus, here you see again the peptidoglycan. The fimbria of these organisms are made up of, among other things, of M proteins. In this instance, not fibronectin, but fibrinogen binds to the M protein, and it stops complement binding. So if a phagocyte is around with a complement receptor, this complement will be bound to the peptidoglycan. In fact, that happens in the test tube. So what happens in this instance is that you'll see the complement as I've drawn it is hanging out here. It isn't attached to the peptidoglycan underneath. So the organism has developed an ability for its surface protein, M protein, to be able to bind the human protein fibrinogen, which blocks the binding of human complement and stops the interaction between peptidoglycan, which would otherwise be covered in complement, because you all remember from the immunology lectures you had that the lipopolysaccharide and the endotoxin-like molecule peptidoglycan can activate the alternate complement cascade. So if it wasn't for the M protein fibrinogen interaction, peptidoglycan would be covered in complement in this instance, and the organism would be taken up. Now, I'm going to actually, this diagram will serve as the basis of a more complicated situation that I'm going to describe in the Group A Streptococcus lecture. We'll have, I think there's going to be 3 different versions of this, so we're going to get into more detail later on. But you've got the basic point. This is what's going on in the human body, and it's what's going on in a non-immune situation. We'll discuss later on what happens in immunity and how the host tries to does its best to get around this situation. As I say, this is a battle going on here. This is 2 armies at work, basically, the bacterial army and the host immune system and other components of its army. And each army is trying any way it can to destroy and neutralize the other one. Okay, returning to intracellular parasites. Basically, 3 things can happen. First of all, the organism might be taken up inside a phagosome. If there is an avoidance of fusion with the phagosome with the lysosome, where all the nasty enzymes are present, such as lysozyme, which chews up bacterial peptidoglycan, if that is avoided, then that organism is perfectly happy inside a phagosome. On the other hand, if fusion occurs, the organism has to be able to actually be able to be resistant to these enzymes, these digestive, because the lysosomes, of course, are full of digestive enzymes. And if the lysosome fuses with the phagosome, then we have a situation here that these organisms have now got to be able to deal with these enzymes and not be destroyed by them. And finally, sometimes these organisms are really smart and they actually get out of the initial phagosome and end up in the cytoplasm. And then once in the cytoplasm, they're away from these digestive enzymes. Okay, so these are the different situations that can ensue. The exact opposite occurs with an interest, whether it's an obligate intracellular pathogen such as Mycobacterium tuberculosis as an example, or some of the other chlamydia will be examples of obligate. Regardless of whether an organism, once an organism is internalized by whatever means, once it's there, it has to be able to deal with cell-mediated immunity, with activated T cells, and the attempts of these T cells to destroy the organisms and the cells that they're in. So that's a totally different situation, because of course antibodies can't get at an intracellular pathogen, it's blocked. Okay, so think back to your immunology lectures. Okay, so now we've got a situation where we've talked about the organisms being on the surface or not being on the surface, being in the air or whatever they are, somehow getting into the internal milieu, spreading, and what have you. But nothing actually what I've really said so far relates to infectious disease, which is really injury. Because I'm not a virologist, but we all know that it's not uncommon to be chronically infected with viruses. And until you get a fever blister or something, you're suddenly aware, oh, they're there. Well, if an organism doesn't do anything, if it's just there, that doesn't make it a disease. I mean, who cares? It might have a little effect on your food intake and what have you. But if it's not doing anything to you, who cares? An infectious disease, by definition, is something you do care about. It's injury. Okay, so all these things that we've led up to at this point relate to the fact of the organism being where it needs to be and being in a position to cause you injury. Now, bacteria are quite smart, and they haven't sat there and said, let me think. Maybe I can find a way that I can kill the host that I'm using for foodstuffs and what have you, and I'll find a way to destroy it so that I can basically die. Bacteria are not suicidal. There are reasons that these things are produced. Oftentimes the exotoxins, for example, we're going to talk about different ways that you can be injured now. But basically these different substances that bacteria produce, they've been produced for one purpose or another. And unfortunately the side product of them being produced happens to be tissue injury or death. And we'll talk a little bit about that as we go along now. But the key point is infectious disease as opposed to just an infection. I can say, well, your skin is infected with the normal flora, for example. But you don't have an infectious disease. I hope you don't. Okay, so that's the situation. All right, so tissue injury, and that's what we're going to focus on now, tissue injury. There are basically three ways that you can be injured in a general sense by bacteria. First of all, the term exotoxin. Now, the word exotoxin and endotoxin are very specific terms. An exotoxin is an enzyme, is a protein that is often an enzyme. I'll show you some definitions on the next few slides. But this relates to specifically a protein, exo meaning outside, being secreted. An endotoxin refers to the components of a bacterial cell envelope, in particular the lipopolysaccharides, the peptidoglycans, the teichoic acids. Endo in this instance means that they're structural components. It does not mean that you're never going to find an endotoxin in the bloodstream. If that were the case, we wouldn't have 200,000 cases a year of death resulting from endotoxemia. What happens is an endotoxin is present as a cell envelope component, and the organism can be chewed up and digested, and these poorly degradable cell envelope components, the endotoxins, can be released. So the endo is not referring to whether the exo and endo are not referring to whether they're inside the cell or outside the cell necessarily. It's referring to particularly their activities and what these things are. And we're going to give you details in a second. And specific immunity is an issue. You've had lectures on immunopathology up to this point. And it doesn't matter if it's autoimmunity or if it's the attempt to destroy a cancer cell, what have you. Anytime you've got a specific immunity to an antigen, the tissue can be destroyed as an innocent bystander. Or there could be other examples of cross-reactivity between a bacterial antigen and a tissue constituent. Because these are the three broad ways you're going to get injury. Let's get into some definitions here. Exotoxins are pretty well always proteins. The key word there is usually. They are very, very often they're enzymes, but they're not always enzymes. I'll give an example of one that isn't in a little bit. They destroy cellular structures, or they can destroy the extracellular matrix. Destruction, that's the issue. And that's what can kill you, and does. An example of an exotoxin. Often they are referred to as AB toxins, and I don't think they were originally defined this way for this reason, but it doesn't really matter. It's often useful to think of the A as being the active component and B the binding component, so you can remember these things. There's usually a component of an exotoxin that recognizes a cell surface or something within inside a cell, and the active component would then be put inside, and it will be or in the membrane or within the cell, and actually destroy the cell. So AB toxins. Most exotoxins are, not all, but most are AB toxins. Two of the most well-known, and I won't have time to spend. There's a lot of information on each slide. Don't worry about it, because every one of these things will be covered again in their individual lectures by me. So here we're just going to make some examples. Diphtheria toxin and Pseudomonas exotoxinae are both produce potent exotoxins. And four of the toxins we're going to talk about carry out ADP ribosylation. That's what's one useful thing from this lecture, is to try and pull together and remember the four that carry out ADP ribosylation. In this instance, these two inhibit protein synthesis, and that's how they kill the cells. Now, you'll notice this one's in this case here, there's two others. Cholera toxin and E. coli label toxin. That's got nothing to do with spinach. We'll talk more about this later on. Cholera toxin and E. coli toxin. In this case, they also AD ribosylate. But in this case, they act on membranes, and it results in cyclic AMP secretion, which in turn leads to water secretion. Okay, so look these over. We're going to go quickly through these now, just as examples. And when you get to the individual lectures, relate what you see in this lecture to those lectures that we cover later on. In the case of enterohemorrhagic E. coli, which is a group of E. coli that produces specific one or several pathogenesis factors, but one of them is sugar-like toxin, and that destroys ribosomes and inhibits protein biosynthesis. These are both Clostridia, closely related species, both act on the nervous system, have absolutely nothing to do with each other in terms of what happens. One produces a flaccid paralysis, that is to say it's floppy, and basically the tissues just don't do anything. On the other hand, the tetanus toxin, which we're also going to cover later, there's overactivity and we get rigid paralysis. Many bacteria can produce the extracellular enzymes that act on the extracellular matrix. Examples here are collagenase that chews up collagen and hyaluronidase that chews up hyaluronic acid. Now this one it's not that it's so important, I just want to emphasize this here. Membrane damaging exotoxins, that is a protease. That fits exactly with everything I've said so far. It destroys proteins. A phospholipase would destroy phospholipids, the two components that make up membranes. Those are enzymatic. On the other hand, if some exotoxins, not many, have a detergent-like action, that's not an enzymatic reaction. And that's why I used the word usually before, because there are a few cases where exotoxins are not enzymes. They actually, in this case, as an example, it's a detergent-like lysis of the membrane of the bacterial cell that causes it to be disrupted. This would be another example of an extracellular, of an exotoxin that did have enzymatic activity, and it's involved in the spreading of Clostridium perfringens. And we'll talk about botulism, tetanus, and gas gangrene in a single lecture, as part of a single lecture, and we'll compare and contrast them. If you have this in mind before we start, it will probably help when we get through into that lecture. Exotoxins are, as I said, proteins. And one thing that's very important, in fact, one of the ways we vaccinate against certain diseases, in fact, the exotoxin can be the hallmark of a disease. It may even be that you don't even have to get an infection. For example, let's take, my mind is going. I'll think of an example in a minute. But anyway, the point is that certain bacterial infections don't even actually get into the bloodstream. All that happens is the exotoxin may disseminate into the bloodstream. And so in that instance, diphtheria is the disease I was trying to remember. In the case of diphtheria, for example, the organism actually lives in the pharynx. And in fact, it lives in the pharynx of normal people. And it doesn't get into the bloodstream. Even when you get diphtheria, the organism stays put and the exotoxin disseminates. Toxic shock syndrome is another example. So the issue comes down to, in a particular disease, if the exotoxin is that important, if you vaccinate against the exotoxin and neutralize it, that should be enough. Well, it is. We don't see diphtheria in this country. And that's what the vaccine is. It's against the exotoxin. We'll again discuss that in more detail later. But that's how important an exotoxin can be. Not always that important. Sometimes it could be that there's a number of different things going on, and one particular exotoxin may not be that important. And it varies from disease to disease, and you need to understand those differences, and you will before the end of this course. Now, an endotoxin, as I said already, is things like, the particular one, the classical one, is lipopolysaccharide. And I don't know if this day and age, really, if one should use the word endotoxin-like anymore. Maybe some people don't. But anyway, peptidoglycan was recognized many years ago to have lipopolysaccharide-like activity. Essentially, anything that a lipopolysaccharide does, a peptidoglycan will do. Now, that means that when you die of septic shock, it means you don't have to die of septic shock just from a gram-negative organism. You can die from septic shock from a gram-positive organism. Because, of course, a gram-positive organism doesn't have a lipopolysaccharide, but it does have a peptidoglycan. So this relates to what I said earlier about knowing whether these things are gram-negative, gram-positive, and what do you have to expect. And that's an endotoxic shock is particularly the most potent endotoxin is lipopolysaccharide. And that's what's causing endotoxin shock in gram-negative infections. And yes, it's not a protein or enzyme. Lipopolysaccharide, lipid and polysaccharide. Peptidoglycan containing non-protein amino acids such as D-alanine, diaminopamelic acid, ornithine, other things that we don't find in proteins. Peptidoglycan is not a protein, and it is not a proteoglycan either. Okay, so these are non-proteins. endotoxins, as opposed to exotoxins, which are proteins and usually, but not always, enzymes. Septic shock is particularly what we think about when we're exposed to endotoxins. And that's the point I was trying to make earlier again, that if you have if the endotoxin that's referring to where the organism has this substance. It doesn't mean that it's stuck there. If an organism gets degraded, the lipopolysaccharides, the endotoxins, can be released into the bloodstream, and they can and do cause severe disease and often death. And this is the sorts of things that happens in septic shock. Typical features of septic: hypertension, disseminated intravascular coagulation, fever, lack of effective oxygenation, and most important of all, overall system failure. Endotoxins are presumably have been exposed to gram-negative bacteria for a long, long time. And it is our job to see these bacteria and our immune system to deal with them. Well, it doesn't always work. And sometimes the organisms grow quite well anyway, and they start producing lots of endotoxins and from the process, or lots of endotoxins were released in the process. These various processes presumably are designed for the host to deal with the gram-negative infection. And here's an example of the host being an innocent bystander, where there'll be non-specific inflammation, cytokine release, complement activation, and you name it. These things have a horrible effects on the immune system. And so that's presumably why we've developed these responses to gram-negative bacteria and attempt to do something with them. It's not, as I say again, that the bacteria are particularly suicidal. Specific immunity, I think this is about 2 or 3 slides now. Specific immunity and immunopathology. Here are examples of diseases caused by cell-mediated immunity. Again, it's not that the bacteria cause cell-mediated immunity and they're doing this to try and damage you. These organisms have found a way to live inside activated macrophages. And no matter how much you activate them, those organisms don't go away. And there isn't even a decent vaccine. It's that hard to get rid of them. So in that instance, we see immunopathology particularly mediated by T cells. There can also not only be persistent infections, there can be persisting bacterial remnants. Proteins are easy to chew up. Peptidoglycans and lipopolysaccharides are not. In the case of peptidoglycans, they can stick around sometimes for years after an infection in people. That's proven experimentally. So the bacterial remnants themselves can cause disease. And the reason that's important is that you can have tissue injury going on, and you do in the case of rheumatic fever and rheumatic carditis as an example, long after an active infection has stopped. You've already had a discussion of autoimmunity, and that's a case where, again, you were trying to deal with the organism, you produced a response, and by bad luck, at the same time your immune response happens to recognize some tissue component of the host. We'll discuss greater details on that when we talk about syphilis, and also when we talk about strep throat. Sometimes organisms will produce proteases to try and destroy the immunoglobulin on the surface. These are particularly organisms that would be infecting through the respiratory route, or in this case through the sexually transmitted organism. So that would be through the urogenital route. But again, on surfaces, you're going to see IgA, which are attempting to specifically destroy particular organisms, and they can chew up that IgA and stop it in its tracks. Finally, bioterrorism. And probably you all know more than you ever wish to know about bioterrorism by now. Air is the most common route. They're particularly from zoonotic infections, I'd say from animals. That's a term we're going to be, there'll be a series of lectures on zoonoses by Dr. Ghaffar, and he will get more into this. Okay, I don't think there's any point making a big fuss about this. This is a bookmark, so when you get to the 3 lectures on bioterrorism, you can relate it back to this slide. Okay, thank you very much. Okay.